Thank you very much, Jonathan. It is a tremendous honor to have been invited to deliver the keynote lecture at the Lomholt Symposium. My work has been made much easier this morning by the excellent preceding talks, and I'm very grateful for all of those. I have no conflicts of interest to declare. So my themes today on the genetic epidemiology of psoriasis include a discussion of pedigrees and the mode of inheritance, then a discussion of what heritability means and how it applied to population and twin studies of psoriasis. These were all very important to me when I was deciding whether or not to pursue genetics as a topic in my career as it developed. So I'm very grateful that things have panned out. So then I'm going to talk just briefly about evolution of GWAS studies, touch again on our most recent GWAS of psoriasis, and then really want to take a closer look at the MHC and the roles of the tonsils in psoriasis. So Lomholt wrote in his thesis, which took a long time to write apparently, question upon question may be asked. The disease is capricious and refuses to part with its innermost secrets. That psoriasis is genetically conditioned is beyond doubt. But when the mode of inheritance appears to have been almost demonstrated, it again slips out of the fetters of fixed rules. You can hear his frustration. And I would just comment that the Faroe Islands don't look very, other than the mountains and the sea, Faroes look a lot different now than they did then. So this is from the International Psoriasis Symposium in 1972.
Lomholt is the first dermatologist assigned to Makerere University in Uganda. You are familiar with his classic study of psoriasis in the Faroe Islands.
One of the things we have recognized is that psoriasis is among the common skin diseases in our part of the world.
He later presented his view that the dominant gene is a genetic basis for psoriasis. But Howard Kahn, in epidemiological studies carried out at Stanford, has a different concept.
Well, I think right now the best explanation for the data on psoriasis is compatible with a determination by multifactorial inheritance.
Dr. Kahn's view is supported by Dr. John Voorhees.
In other words, I think there's a very poor possibility that a single gene, in other words, a single enzyme, or even one or even two or three, will turn out to be the ultimate answer in psoriasis. I think there are multiple alleles involved. And I think that any study of psoriasis should take this into consideration.
We have all learned from each other at this symposium and share the conviction that we must work together to solve the problem of psoriasis.
So I want to talk a little bit about some pedigree studies, which was really generated some of that discussion that we heard about in the film. We have already heard about Lomholt's pedigrees, and there were many of them. And so I want to focus on a large family that was published by Abela, Dobson, and Graham in 1963. Actually, for people who want to learn the earlier history of pedigree studies, that has an excellent summary of the earlier studies. This was a North Carolina kindred containing 815 Caucasian Americans spanning 6 generations, and the pedigree is shown on the right. And it's really interesting that this pedigree was later collected by Ann Bocock with support from the National Psoriasis Foundation and formed the basis of the discovery of CARD19 as the SORAS2 gene in 2012. And subsequently, CARD19, sorry for the typo, variants also were shown to underlie pityriasis rubra-pilaris. So, since we've already heard about Lomholtz studies, I will move on. So a concept that I want to discuss here briefly first is called heritability. It's abbreviated H squared. And this is Falconer's model of disease liability, where factors underlying liability are basically normal distributed or a bell curve, and people with liability factors above a certain threshold will be affected, in this case by psoriasis, but for any disease. And as we're going to see, the component of the heritability is the component of variation that is due to genetic factors, presumably the remainder being due to environmental factors. So, not only was Lomholtz a great pedigree study, but it was also really a population study where everyone in those 7 islands was studied. And I stand corrected, it wasn't the 7 biggest islands, it was the 7 northeasternmost islands. Now, Anantha Christian and al. In 1974 analyzed those pedigrees and determined that H squared could be calculated at 91%. That turns out to be kind of on the high end. In Hellgren's population study in Sweden, you can see the prevalence was higher in first-degree relatives of psoriasis patients, also higher in second-degree relatives than in normal controls, but not matched controls of relatives of normal patients matched for all the same factors, but certainly higher than normal controls in general. And using these data, the same group determined H squared to be 64% for parents, and there's a typo here, 60% for siblings of psoriasis patients. So that you can see where this sort of range of heritability is shaping up. So then another way to calculate heritability is twin studies. Brandrup in 1978, studying the Danish twin registry, found that 68% of monozygotic probands had a psoriatic twin. And that, again, yielded a heritability of 91%. Now, Farber found a similarly, he was studying retrospective, basically from the literature, twin studies. So it was not a population study by any means. But they found basically the same higher rates in monozygotic twin pairs of concordance than dizygotic twin pairs. Now, the next study listed here is from Duffy in 1993. This was from Australia. And again, the concordance rate in identical twins, 35%, and versus 12% in dizygotic twins. And this also yielded a high heritability of 80%. So both of these, percentage rates are lower than have been in other studies. Part of it could have been that their patients were younger, but I've always been intrigued by the fact that it might be lower because ultraviolet light could be an environmental factor. That was never really proven. So a study by Grzybalski in the Norwegian twin panel of 8,045 individuals, 4.2% reported having psoriasis and no sex difference. The monozygotic twins were more likely than dizygotic to develop psoriasis if a co-twin reported having psoriasis. And while they didn't calculate heritability exactly the same way, they used an additive genetic model and showed that the best fitting model was additive, and it could explain 66% of the variation in heritability. Perhaps the most striking was in all 4 of these studies, among the monozygotic twins, there was striking similarity across twins in the age of onset and the manifestation of the disease. So that it really indicates that while multiple genes may participate, is when you basically, in two identical twins inherit the same genes, they're going to end up with the same clinical manifestations. They're not all the same. There are quite a few different clinical manifestations when you look across all the twin pairs. But among the concordant identical twins, they were usually very, very similar. So when I came to Michigan, I didn't know whether I wanted to study psoriasis, but it turns out that Francis Collins, former director of NIH, was with me as a grad student when I was a PhD student in Sherman Weissman's lab at Yale. And he moved to Michigan as well, where he developed a program in human genetics, initially focusing on monogenic diseases like neurofibromatosis or breast cancer. But he was very interested in polygenic disorders, and at that time, I had just read the textbook chapter by Harvey Baker in Rook's Textbook of Dermatology, which was an excellent chapter and discussed all the evidence for heritability of psoriasis. So I went to Francis and I said, don't you think if you want to study polygenic diseases, wouldn't psoriasis be a good one? And he said, yes, it would. Why don't you write a pilot grant for my new genome center? And I did, and that's how it all started. So we've heard about the genetic epidemiology studies. I want to touch a little bit on HLA associations. These are done in a case control format. And, it took quite a while for the HLA system to be worked out and understood. We all take it for granted now. But in the 70s and 80s, the structure of the genes in the HLA complexes and how they broke into class 1 and class 2 genes had become known, and there were typing assays that could be done. They didn't involve DNA, they involved serology, but they worked reasonably well. And psoriasis was one of the first diseases to be studied using these techniques in 1974. Multiple sclerosis was the first, and it was class 2 associated. Ankylosing spondylitis was the first class 1 associated disease, psoriasis following immediately thereafter. So in the 80s and 90s, we had microsatellite markers. These markers were not as frequently spaced throughout the genome as today's SNP markers are, but they were highly polymorphic, and they were suitable for tracking tracking alleles through families. So the idea is you have a set of markers and you ask which markers co-transmit through families with the trait. And this works well in situations within families, but it turned out not to work quite as well for some genes. So a parameter called lambda S is a parameter that compares the percent or the prevalence of psoriasis among siblings versus the general population. So you can see that if the population estimate is 2%, then lambda S is 5. If the population estimate is 1%, lambda S is 10. And Risch and others showed that if lambda S is greater than 4, and there's a strong gene effect, that linkage will be detectable. And for psoriasis, depending on the population estimate, in either one of these estimates, lambda S was greater than 4. So that was encouraging. So these are our first 22 families studied for a microsatellite marker near the CRP locus. Now, this locus is really close to the epidermal differentiation complex. And you can see as we started, it was quite laborious to collect these families. As we started getting them, we were so excited. 3 is the gold standard for linkage, and we were just almost there. And then it turned the corner, and we were back out of luck. So this was, not only this type of problem was found by people in many diseases besides psoriasis. But I'm going to go back a slide now, 2 slides. And I'm sorry, I should have detailed this, but if you follow the yellow chromosomes in this panel, you can see that a particular HLA haplotype is segregating, and that has HLA CW6 in it, is segregating through this pedigree, but there's a couple of other chromosomes that have CW6, but they have other flanking markers. They were brought in by unaffected people. And they can end up in the next generation of people even if the original chromosome wasn't there. So this is an example of association and linkage. And we wrote papers where we looked for evidence of linkage in the presence of association for MHC haplotypes, and it was a very, very strong result. So that means, so this is, I'm going to show you biologically too, but this reflects the fact of how major an effect that HLA has on psoriasis. So with the turn of the millennium, genome-wide association studies, or GWAS, became possible with the advent of single nucleotide polymorphism markers. There are many, many, many more of those across the genome, so they're much more dense. So essentially, they can map meioses going back for all of human history, and give you much higher genetic resolution. In the bottom panel, while it's small, you can see that very big peak for HLA complex. And Michael has already shown how big it is. So I think everybody here is familiar with what is measured in a GWAS. It's differences in allele frequencies, and sometimes those differences are very small, which is why big numbers are needed to make GWASs work. In the MHC, the effect was large enough that's why associations could be detected so long ago, because the effect is so big. You can see the difference between the blue bar and the yellow bar for HLA-CW6. So this is our expanded meta-GWAS. You've seen this slide before. I'd like to thank not only Michael, but Nick Dand, Alex Soy, Rajan, and Phil Stewart for working on this big study. Many groups, including many here, were involved. And we have 109 LD blocks with one or more independent loci. There's 148 non-MHC loci. 27 of them have more than one independent signal. 2 have more than 3. The MHC actually has 17. That comes from a different study. This study could not study the MHC because we only had summary statistics, and you can't look for secondary signals effectively when all you're dealing with is summary statistics. We reproduced all but 2 of the previously reported susceptibility loci, and so we were quite happy with that. So I've used this slide for many years. Psoriasis, a play in 4 acts. Johan has just elegantly summarized a lot of this, much more elegantly than is shown here. But today I want to focus on the MHC component and also the role of proteases. So this is a study from Hensler and Christopher's study of type 1 psoriasis, and showed, this is data from their study, which shows the percentage of people with psoriasis, parents, children, and siblings, in the so-called type 1, which is young age at onset, typically HLA-CW6, often severe disease. And that's not a great criterion. It was, it's not what we should be using today. And type 2, which is onset greater than 40 years, you can see how much lower the percent of people with psoriasis is. So this paper was published last year in Human Genetics and Genomics Advances. And the top panel is people of South Asian ancestry, a much smaller study, about 2,000 cases, 2,000 controls. Much larger study of European ancestry subjects, and the bottom panel is with them combined. And you can see the shape of these curves is very similar. And that really reflects the fact that the history of people in South Asia, most of them have similar mid-distant ancestry with Caucasians. And so it wasn't surprising that their HLA association curves come up the same. And there are, like as I mentioned, this study found 17 independent loci in extended MHC when the 2 populations were combined. Only 2 of the loci in the final model were protein changing, and one of them was HLA-CW6. The other is position 67 of HLA-B. 4 other signals did occur in genes, but weren't protein changing. And regulatory variants were also found. Most of them were regulatory, and they, by using established databases, they also appeared to affect the expression of class 1 genes much more than class 2 genes. So one of the take-home message is these signals seem to reflect variation in HLA class 2 and not some linked gene, and second, that it can affect both the allelic structure and the regulation. So Johan already touched on this, but the panel on the left is an example of what's called epistasis. So that is the odds ratio for ARAP in the absence of HLA-CW6, it's quite low. But in the presence of 1 copy or 2 copies of HLA-CW6, it's higher. And that's a classic example of epistasis. And ARAP1 signal is actually relatively quite strong in our GWAS. And on the right, it shows what ARAP does. It's a protease that cuts off the end of peptides so that they can fit in the MHC. And I like this picture. It's sort of taking a hot dog, a long hot dog, and trimming it down so that it fits inside the MHC class 1 bun. I always like this one. And, you know, it's not just psoriasis, but also ankylosing spondylitis and Behçet's disease, which are the other 2 major HLA class 1 diseases. Both show ARAP1 associations. And so this seems to me to be just really strong evidence that active antigen presentation via MHC class 1 is central to pathogenesis of the disease. This is another slide that it's really old, but I love it. This is a study from Jörg Prinz where they got tonsillar T cells and skin and blood from psoriatic patients. They did TCR spectrotyping. And so when you look at blood T cells, there's multiple peaks, meaning polyclonal T cells, no expansion. Total tonsillar T cells, not much there. CLA negative or non-skin homing tonsillar T cells, not much there. CLA positive T cells, now you see expansions of specific T cells. And as you can see, they all seem to line up with peaks that are found in the skin. And you know, the tonsils are a place where basically you're developing immunity that's supposed to be present, you know, in the tonsil itself and in the oropharynx epithelium. And these are breeding grounds for skin homing T cells. And the fact that the pharyngitis of strep usually precedes skin lesions means that they're more likely to be pathogenic than to just be innocent bystanders. But the antigens remain incompletely understood. So, you know, there's long been known this strong association between guttate psoriasis, strep pharyngitis, and HLA-CW6. And that's just been found repeatedly. Now, so how did it happen? Helgi Valdemarsson, who trained Johan and also Andy Johnston, was strongly believing in a cross-reaction between peptides from keratin 6 and 17 along with strep M proteins that are both filamentous. And Lionel Fry also thought that strep peptidoglycans might serve as natural adjuvants to get the reaction going. Now, more recently, I don't know if many of you have seen this, Graham Ogg at Oxford has long been interested in CD1A, which presents lipids via a class 1-like molecule. So what he did is he set up a model in which K562 cells, which lack antigen presenting activity, could be transfected with CD1A, so they could present lipids. And then he actually infected them with strep. He had to fiddle with the conditions so not all the cells died too quick. But when he found the right conditions, and then he looked at the T cells coming from normal people, that 5 to 10% of normal human T cells respond to strep pyogenes in a CD1A dependent manner using this model with the strep infected K562 cells as the starter. So Michel Gillier has made a story about LL37. There's a related story that LL37 protects nets, and it protects them from attack by strep proteases. So there may be a strep and staph too. Maybe a connection there. One that where isn't much of a connection is Jörg Prince's careful study where he presented peptides to T cells by HLA-CW6 and found using peptide libraries that this peptide from ADAM-TSL5 expressed by melanocytes could get the job done. Now, how that relates to strep, I can't say, but having an antigen from melanocytes would certainly help to understand skin specificity. And this comes from actually Johan and his team. They did a controlled trial of tonsillectomy for psoriasis. So what you can see, what they did is they had 29 patients, and they all had a documentation of history of exacerbation after sore throat. So this isn't all comers, but they're people who were known to have a problem with strep. And they were either given tonsillectomy or not, and then monitored for 2 years. And they actually monitored skin-homing T cells that responded to peptides that are homologous between M protein and keratins. And 86% of patients showed sustained improvement after tonsillectomy, similar to Enbrel, actually, in terms of how well it works. And then there was close relation between the degree of clinical improvement and the reduction and frequency of peptide-reactive skin-homing T cells. And that is shown here. So the upper group is the tonsillectomy, and the bottom is the controls. So what about, you know, when you say, you know, what's the biological role of the MHC, I think that whatever it is, you can interdict it by taking out the tonsils. Lots of other things affect the tonsils other than strep, but strep is certainly a biggie. So I think that's something that we can take to the bank. Now, all I did with this graph is turn it sideways from the way that Johan had it. The genes across the top are genes that fell out of a TWAS, or transcriptome-wide association scan. It means that their regulation is affected by psoriasis-associated genetic variation. And the vertical axis is all the different cell types. And I know it's too small to read, and we've already talked about it, and I'm running out of time. And it nominates candidate genes from very much different cell types. And we had, Johan already went through this about the clusters that seem to work, so I won't belabor that. And this is also the figure that Johan already showed you involving cytokine-stimulated keratinocytes, showing that the biggest overlap with psoriasis TWAS genes is when these keratinocytes were stimulated with IL-17 plus TNF. Now, you know, in the recent years, and I'm not really going to talk about this, but I made the fateful decision to try to understand the nature of these genetic signals. Most of them are regulatory, and so they don't make a detectable change in gene structure. And we don't really have a complete lexicon for understanding how sequence variation affects transcription. So we sort of set out to start to do it ourselves. And we know that sometimes the genetic signals are far away from the genes they regulate, but often they're close. And linkage disequilibrium is a major, major challenge. And we need these tools, so we decided to try to develop them. Now, when epigenetics are the focus of investigation, the cellular system you use makes a difference for many genes. And what we decided to do, for better or for worse, is use CD3, CD8 stimulated PBMC, which is a very crude system for this purpose, but it does robustly support Th17 expansion. And we'll be presenting data in future meetings about this. So to summarize, Blomholt was right, even though he wasn't completely right. The major, you can think about the MHC as that major gene that he thought was true. But there's a whole host of of minor, I wouldn't call them minor, but less, genes of lesser effect scattered throughout the genome. So Kahn was right too. So I'm glad that everybody gets to be right. And so genetic epidemiology studies of population and twins do support multifactorial inheritance. We have an expanded GWAS of psoriasis that's doubled the number of psoriasis loci. The MHC remains the big kahuna with at least 17 independent genetic signals within it, most of which regulate either all of which essentially regulate either the structure or the function of the HLA class 1 genes themselves. And there's abundant evidence for the importance of the tonsils as a key link between genetics and the immunology of psoriasis. And our future work is to try to pinpoint the variants underlying these genetic signals using both RNA-seq and single-cell RNA-seq, as well as ATAC-seq as a probe and chromatin structure. So with that, I'd like to thank my many contributors over the years from the University of Michigan, particularly Alex Soy, and who is here, Johan, who is here, Rajan Nair, and John Voorhees, who've been very, very supportive. The group in Kiel, our biostats team, the group, Soumya Ray Chowdhury, who was important in the psoriatic arthritis story, all the people in Canada from IPART, all the group from King's College London, and so many others, and I'd like to thank my sources of support. So please contact me if you have any questions, and thank you for your attention.
JT, that was an absolutely fabulous talk. Thank you very, very much indeed. We do have time for some couple of questions, if that's okay. Mahira, first with you.
Thank you very much. Amazing talk. I have two questions. The first pedigree you showed, they had this symbol for carriers. How would they know that? And they had this question mark beside it, the older pedigree. How would they know they were carriers? I was just curious about that.
Well, because they were unaffected, and they were modeling on a dominant condition, okay, not polygenic.
So probably
They inferred it. They inferred it because they had offspring.
yeah.
They had offspring who were affected, and the parent was not.
The other thing about the tonsils, I mean, are you talking about the tonsils only in terms of having a strep infection or as an immunological organ? Because studies that were done about tonsillectomy, even in children, they were inconclusive. They were not 100% sure that they really improved psoriasis, even psoriasis proved to be caused by a strep infection. So I was just wondering about that.
You nailed it. It's the latter,
Yeah.
because Johan and Helgi's study pre-selected for people who had evidence of documented triggering by strep. So no, it shows that the tonsils. The study shows that the tonsils are important in that selected group of people. Yeah.
Question about Robert.
When were you able to find that amazing video from 1972?
Yeah.
Are you ready for this? Eugene Farber gave me a copy, and I still have it. And I went over to the video center and copied pieces of it over to make a file.
Maybe we should see if IPC can host that one as well. That would be quite fun.
I have it.
Any other questions? JT, what's happening in people who don't, who are HLA-CW6 negative, and presumably the other HLA signals as well?
Well, I mean, according to NO and TILO, they probably have older age of onset. And as we heard about in drug responsiveness, that the people who were CW6 negative tended to be, responsive to anti-TNFs, but not to anti-IL-23. And so I would say that as people get older, that maybe it's HLA alleles, maybe it doesn't even have anything to do with HLA. That is enough to get the condition rolling. But yeah, the role of the other 17 independent signals is just mind-boggling, and actually is kind of scary.
Back to the tonsils and the strep story. If you look at the genetics and the type 1 interferon, there seem to be so many genetic signals suggesting antiviral pathways. Do you think it's strep or, I mean, tonsillitis is usually viral?
It certainly could be. It certainly could be. And I'm glad you brought that up. The whole thing about where do these responses to, why are so many receptors for double-stranded RNAs, why is that, is fascinating. But I think you make a really, really good point, Jonathan.
Andy?
At one point, there was some literature about retroviruses being presented as class 1, possibly linked to psoriasis. You didn't mention any of that. Has that been all disproving?
People tried to replicate some of that work, and it didn't fly. I'm not going to say it couldn't be. I mean, basically, you know, the body has its own ways of sorting out our own cytosolic RNAs from those that come from viruses, involving the ends of the molecule and their double-stranded configurations. And it, you know, any, I suppose any virus that could, you know, fool that system could set off the alarm.
Ruth?
Thank you. Lovely lecture. There's so much warmth and wisdom. I just wondered, you know, the immune pathway to psoriasis is so elusive and there's so much redundancy. Is there a selective advantage for having psoriasis?
That has actually been considered. I believe it was Richard Chalmers, or who actually found, well, that basically, you know, the strep killed a lot of people in the past, and the people who mounted a more vigorous immune response to group A strep may have had a selective advantage. I don't know if that's the right author. I'm sorry.
I'm not sure who that was.
Okay.
Was it Lionel Fry?
Lionel Fry. Yeah.
Okay. Well, let's stop on that note. And JT, that was a fabulous talk, and we appreciate it very much, and lots to think about over lunch. So thank you very much.
It was an honor, and thank you all very much.